students, how are you all? I'm very happy to meet you all. May Almighty's peace be upon you. During this COVID-19 time, it's our duty to prevent us from COVID-19. I have told you some of the drinks and what you have to eat in my previous videos. But before that, understand, staying safe at home is very important. Social distancing is very important. So now the COVID-19, the coronavirus spreads through air and it is our duty to wear the mask. Wear the mask anytime you go out, everywhere you go. My dear students, in my last video class, I was teaching you about pollen grain, pollen sac, tears of mature anther, how the male gametophyte is formed. So in this video, you will be learning about the female gametophyte, the female part. Yes. So when we speak about the female part of the flower, it is gynoecium. Gynoecium, the female part of the flower. So when we speak about gynoecium, so we say it as pistil. Pistil. So in a flower, pistils will be there. Pistil is made up of carpels. Now, so pistil. So three parts are there in a pistil. One is stigma. The second one is style. Third one, ovary. So stigma, style, ovary. Three parts in a gynoecium, in a pistil. So what are they? How do we know it? So generally, you would have seen. So you imagine that this is stigma. You will have some colored part, some velvet colored in hibiscus. That is your stigma. So this portion, stigma. And the filament of the gynoecium, the filament is called as style. Filament is called as style. And the most important part, ovary. Now, again, we have to learn something in detail about these parts. What is the role of this stigma? Stigma, it is the landing portion of stamens. Landing portion of stamens. We already said that pollen grains will be liberated from anther and then the pollen grain will reach the stigma. Only when it reaches stigma, it absorbs moisture, swells up, then a pollen tube is formed. So this portion here only, the pollen, when the pollen comes, this is the landing portion of this stem, landing portion. Stigma is the landing portion. And style, again, it is the long tube, the filament of this, and here only the pollen tube is developed. When it is mature, the pollen tube travels through this style. The third part, ovary. Ovary, it has ovules. So if you see, the ovule is present here. I am drawing a rough diagram. So ovule is present. So this everyone is ovule. So ovary is enclosed in ovary. Ovule is enclosed in ovary. Now this one here, we have so many ovules, isn't it? But in some cases, you will have only one ovule, right? Like rice, wheat. So in such plants, we see only one ovule. But in some, there are many ovules are also present. So this attached portion, no? This is called as placenta. Placenta, you would have learnt in 11th standard about placentation. So placenta, the attached portion. This is the general structure of gynoecium. So here the carpels also will be different. That is, the number may vary from 1, 2, 3, 4 likewise. If it is 1, that is called as monocarpellary. It is just to recall what you learnt in 11th standard. Monocarpellary means it will have only one carpel. Bicarpellary is there. Bicarpellary. Two carpels will be there. Tricarpellary. Musa paradisaica. Banana is a tricarpellary plant. Then even polycarpellary. Many is also there. If you see hibiscus rosa sinensis, it is pentacarpellary. 5. So accordingly, the ovules, everything will be 5. It will be in 5. So this is the general structure, the basic thing you should know about gynoecium. And hope you remember about syncarpus and apocarpus. Syncarpus, when the carpels are united, that is syncarpus, and when they are free, it is apocarpus, which you can see in aggregate fruit. You would have learnt in aggregate fruit, polyalthia.
The gynoecium represents the female reproductive part of the flower. It represents one or more pistils of a flower. It has stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is an elongated, slender part beneath the stigma where the pollen tube grows. The basal swollen part of the pistil is the ovary. The ovules are present inside the ovary cavity, called as locule, on the placenta. Gynoecium arises a small papillate outgrowth of a meristematic tissue from the growing tip of the floral primordium. The ovules are also called megasporangia, which arise from placenta.
Now, if we see the structure of ovule, we have said that the base swollen part of gynoecium is ovary and ovule is enclosed in ovary. And now we will discuss about the structure of ovule. So this ovule, when we speak about, ovule is said to be megasporangium. Megasporangium. You remember which is microsporangium? In androecium, when we were speaking about pollen grains, the male gametophytes, we discussed about microsporangium. This is megasporangium. This is bigger than our male gamete. So megasporangium. Ovule is also said to be megasporangium. Now let's discuss about the structure of ovule. I know very well that you would have studied it in your 10th standard. So actually the diagram given in your book is inverted. Okay, but as we are following the same, let's go through the same thing. Now, the main part of the ovule, okay, the main part of the ovule is said to be nucellus. The main part is nucellus. The main part of the ovule. This ovule is covered, this nucellus in ovule, this nucellus is covered by the layers. So one or two layers. So covered by layers. So these layers are said to be integuments. Integuments. Okay. So integuments. So this is ovule. These are the outer coverings called as integuments. So again, based on how many integuments are present, it is classified as unitegmic or bitegmic. Unitegmic means only one layer will be there. Bitegmic means two layers will be there. Now, so this at the top, as I said already, this is an inverted diagram. Okay. So at the top, this side, okay, this integument, there will be a free space. That is here nothing. There will be an opening. There will be an opening at the top. This opening is said to be micropyle. Micropyle. Okay, micropyle. And this ovule. Ovule is connected to the placenta. This ovule is connected to the placenta by a stalk called funicle. Funicle. By a stalk called funicle. So funicle. Stalk called funicle. Where the funicle attaches this ovule. Okay, where the funicle. So this place, this is said to be hilum. Hilum. So where the funicle attaches with this ovule, this portion is called as hilum. Okay. So the main portion is nucellus. It is covered by layers called integuments. There is an opening is called micropyle, and the stalk which is attached to the placenta and the ovule are attached to the stalk called funicle. Where the funicle attaches the ovule, that position is placed is called as hilum. Now opposite to micropyle, this portion, okay, this portion, this is called as chalaza. So in an ovule, we say this as micropylar end and this as chalazal end. Micropylar and chalazal end. Okay. Now, so here in this nucellus there is an embryo sac. Embryo sac is present. Embryo sac. In this embryo sac we have 8 cells. Okay, that we will discuss in next class actually. So 8 cells will be present. So 3, 2, 3, 8 cells. All these are made up of parenchyma cells. Totally parenchyma cells will be present here. This is the basic structure of L.S.of ovule. If you see the longitudinal section of ovule, you can see these parts. Okay. So as I said, towards the micropolar end, there is a sac called embryo sac present, isn't it? So embryo sac is present here. Now, in some plants, in some species, the inner integument supplies nutritive tissue. So the cells present here in inner integuments, which gives nutritive tissue to the embryo sac, they are called as endothelium. So endothelium cells are integumentary tapetum. You remember tapetum gives nutritive tissues, nutritive for the anthers. In tears of mature anther you would have learned. Here endothelium is the special cells present in the inner integuments which gives nutrition to the embryo sac. So based on the sporogenous cells present in the nucellus, okay, in this region, the type of cells present, the ovules can be classified into 2. One is tenunucellate and another one crassinucellate. So this tenunucellate is nothing but here this layer, it will be made of a single layer of cells. Only single layer of cells will be present here. In this hypodermal region, only single layer of cells are present. But in crassinucellate, you can see double layers, more than one layer. So the layers will be like this. You can see after this, only one layer will be present here, but here 2 layers will be present. 2 layers. So this is tenunucellate and crassinucellate. 2 types are there. Then finally in ovule we should know the cells present near the chalaza. The cells present near this chalaza, they are called as hypostase. Hypostase. And cells near the micropyle is called as epistase. Epistase. So hypostase, epistase. So 2 types of cells are found in ovule. So my dear students, this is the basic structure of L.S.of ovule, and based on the type, how the funicle, everything, there are different types. Before that, let's see the picture of L.S.of ovule.
The body of the ovule is made up of a central mass of parenchymatous cell called nucellus, which has large reserve food materials. Ovule is protected by 1 or 2 coverings called integuments. A mature ovule consists of a stalk and a body. The stalk or the funiculus is present at the base and it attaches the ovule to the placenta. The point of attachment of the funicle to the body of the ovule is known as hilum. In an inverted ovule, the funicle is adnate to the body of the ovule, forming a ridge called raphe. The ovule with one or two integuments are said to be unitegmic or bitegmic ovules. The basal region of the body of the ovule, where the nucellus, the integument and the funicle meet, is called chalaza. There is a large oval sac-like structure in the nucellus toward the micropylar end called embryo sac or female gametophyte. In some species, the inner layer of the integument may become specialized to perform the nutritive function for the embryo sac and called as endothelium or integumentary tapetum. Based on the position of sporogenous cells, it is classified, ovule is classified as tenunucellate and crassinucellate type.
Ovule is classified into major 6 types. So major 4 types plus 2. So 6 types based on the position of micropyle, funicle and how they orient. So depending upon everything, so they are classified into major 6 types. Okay, let's discuss one by one. First type is orthotropous. Orthotropous, it is seen in Piperaceae family, that is our pepper family. So Piperaceae family, what happens here, the funicle will be here, so you can see, and the micropyle will be present here, micropyle, chalaza here. So totally everything in the same straight line. So everything in the same straight line, it occurs in same straight line, that is orthotropous. So micropyle, funicle, micropyle, chalaza, together all the ovule will be, the embryo sac will be here, together everything will be in the same straight line. That is our orthotropous type of ovule. Second one is anatropous. Anatropous is seen in most of the dicots and monocots, where generally we saw the diagram of our L.S.of ovule, no? How we saw. So there are micropyle, integuments, likewise. So this type of, so micropyle will be here, chalaza here, funicle. So this type, okay, generally this anatropous is nothing but micropyle and funicle will be lying close together. That is the anatropous, which we saw in detail the structure of L.S.of ovule. So micropyle and funicle lie close to one another. That is anatropous type of ovule. Third one is hemianatropous family. Primulaceae family, our herbaceous plant, garden plant, primroses. So primroses, that family actually. So here what happens, the funicle will be here and it will be held, the micropyle or ovule, everything will be held at right angle. That is, the ovule will be present like this, totally. So micropyle will be present here, micropyle will be here, embryo sac will be here. So this is both the funicle will be present here. So they held at right angle, that is said to be hemianatropous. So you remember orthotropous and totally straight line, but here hemianatropous, right angle, held at right angle. Finally campylotropous. In campylotropous, it occurs in Leguminosae family. Here B-shaped legumes, generally P varieties, isn't it, legumes. So here in this, if you see, the ovule itself occurs as bean shaped, somewhat curved one. The ovule, the inside the embryo sac will be somewhat curved one, totally it will be curved one. The funicle will be present here, the micropylar end, the embryo sac will be present somewhat like bean shape. So the ovule will be present like this. So I am drawing rough diagram only. I will show you the real picture after this class. So they occur as bean shaped, and the embryo sac is slightly curved also here. And if you see the hilum, micropyle, chalaza, everything together only, because it's curved one, isn't it? So everything is curved. Then the next type is, the next two types are there. Apart from these four types, next two types are there. Next two are amphitropous and cercinotropous. Actually, amphitropous is nothing but here the hilum and chalaza are held together. So totally embryo sac will occur like a horseshoe type. So it occurs as horseshoe type. Micropyle, here hilum. So chalaza is present here. Actually, hilum, chalaza will be at the lower end of the ovule actually. The opposite to micropylar end. But they will be held together. Okay. And the embryo sac appears as horseshoe type. So this is amphitropous. This is seen in aquatic plants like L.S.of, aquatic plants. Cercinotropous is here the funicle it engulfs or it covers totally the ovule. So ovule. So here micropyle is here. Totally it will cover. So actually funicle will be in a less portion only it will be. But here what happens? The funicle covers the total ovule. So that is cercinotropous. This is seen in Cactaceae family. Cactus family. Our xerophytes. Cactaceae family. This is seen in aquatic plants. So totally 6 types of ovules are seen in plants. Orthotropous, anatropous, hemianatropous, campylotropous, amphitropous and cercinotropous. So again, orthotropous, everything lies in straight. The funicle, chalaza, micropyle all lie in a straight line. Anatropous, our normal one. That is the funicle and micropyle will lie in close together. Hemianatropous, they are held at right angle. And campylotropous, again our hilum, micropyle, chalaza will be close together. And the amphitropous, again the hilum and chalaza are together, and the embryo sac occurs like horseshoe. Then cercinotropous, the funicle encloses the ovule, so it covers total ovule. So these are the types of the ovules. Based on it, we have so many plants. These types of ovules are seen.
In orthotropous type of ovule, the micropyle, the funicle and the chalaza lie in one straight vertical line. Examples: Piper family. In anatropous type of ovule, the body becomes completely inverted so that the micropyle and funiculus come to lie very close to each other. This is found in dicots and monocots. In hemianatropous type of ovule, the body is placed transversely and at right angles to the funicle. Example: Primulaceae, primrose family. In campylotropous type of ovule, the body at the micropylar end is curved and more or less bean shaped. The embryo sac is slightly curved. All the three, that is hilum, micropyle and chalaza, are adjacent to one another with the micropyle oriented towards the placenta. Example: Leguminosae. In amphitropous type of ovule, the distance between hilum and chalaza is less. The curvature of the ovule leads to horseshoe-shaped nucellus. Example: aquatic plants, Alismataceae. In cercinotropous type of ovule, funiculus is very long and surrounds the ovule. Example: Cactaceae, xerophytes. Assignment questions. Number 1. Write a sentence for A. Nucellus, B. Hilum, C. Chalaza, D. Embryo sac. 2. Draw the structure of L.S.of ovule. 3. Write a note on endothelium. 4. Explain the types of ovules based on the position of sporogenous cell. 5. List the types of ovules based on the position of micropyle and chalaza. Explain hemianatropous and amphitropous.